Hi there, my name is Claire Baiocchi, and my research with Dr. Drew Roll related to selecting antibiotic-producing bacteria from soil using different concentrations of tetracycline. So when we're talking about antibiotic-producing bacteria, it's important to note that they are likely to be resistant to multiple different antibiotic types. And that's just a testament to the diversity of the microbial community that's in the soil and the increased use of antibiotics that is kind of sort of backwashing into sewage and wastewater and ultimately affecting our soil as well. So it's an important issue to select for and find different antibiotic producing and antibiotic resistant bacteria in the soil. And my project related to increasing the efficiency of selection for antibiotic producing bacteria and seeing how many different types of antibiotics are these bacteria resistant to. So I did a set of serial dilutions with different concentrations of tetracycline, 3 micrograms per milliliter and 30 micrograms per milliliter, and both of those had 10% TSA, and then just a regular 10% TSA control dilution set. And then what I found was I did have growth consistently on 30 micrograms and 3 micrograms, less so typically on 30 micrograms per milliliter, which makes sense. And then I would isolate these, the colonies from the dilution set further and do antibiosis testing against 3 different soil microbes, Escherichia coli, Bacillus subtilis, and Staphylococcus aureus. And what I found was that I did about 24 samples from my data set, and what I found was that there was the highest amount of antibiosis in Bacillus subtilis, which makes a lot of sense because Bacillus subtilis is a very common soil microbe. And now I knew that these antibiotic producing bacteria that could grow on tetracycline, so that means something. They can produce antibiotics. Now I want to see how many different types of antibiotics they're resistant to. And then I did a Kirby-Bauer test with only 6 samples. So this was toward the end of my research. I didn't save all of my other isolates, but what I did find was there was a diverse amount of antibiotic resistance against gentamicin, chloramphenicol, imipenem, penicillin, ampicillin, erythromycin, and streptomycin, which are all very common antibiotics used in clinic. And the highest was in penicillin and ampicillin, the beta-lactam antibiotics. However, I can't really make any claims because the data was limited and small. So, but overall, my project and my hypothesis with Dr. Roll supported the idea that we can increase the selection for antibiotic producing bacteria in soil by using different concentrations of tetracycline. All right, thank you.